Assalamu alaikum everyone, how are you? Inshallah everybody is doing well. So this is the presentation for our lesson 2. Okay, so in this lesson we are going to talk more about codominance and incomplete dominance, because we have so far seen traits or diseases which are dominant and recessive, but this is not the case for all traits or diseases. So we are going to see some examples here. So we will explain what is codominance and incomplete dominance in the inheritance of human blood groups and sickle cell anemia. So this is two examples where we are going to see where you have this incomplete dominance and codominance, and using diagrams to predict inheritance. Okay, so there are some characteristics which doesn't follow Mendelian inheritance, means they are not this kind of recessive and dominant. So they call it non-Mendelian inheritance or non-Mendelian characteristics. So I put here the picture of Mendel and the stop sign just to show you that there are some characteristics which doesn't follow Mendelian's inheritance. So here some traits don't follow the simple dominant recessive rules that Mendel first applied to genetics, and traits can be controlled by more than one gene. So here you have to know that Mendelian inheritance or dominant recessive genes usually are controlled by one single gene, or that character or trait is controlled by one single gene. But this is not the case for many traits, like for your skin color, even for eye color also, it's not controlled by only one gene. So in this case you cannot really see dominant recessive rule. So these traits can be controlled by more than one gene. Okay, so some alleles are neither dominant nor recessive. And you have the examples here. For example, in few animals you can see different colors, like Dalmatian dogs. You can see black and white. So here the black is expressed, the white is expressed. So both of them are expressed. So the black is not covering fully the white, and the white is not covering fully the white. So they are called codominant. In certain cases, like here, for example, if in certain flowers, if you mix the red flower with the white flower, it will give you an offspring which is pink. Okay, this is not the case for the pea plant. The pea plant we have seen before. So this is other plants, they have this trait actually. So this one is called incomplete dominance, where you have a mixture, okay, of traits. So here you have incomplete dominance or lack of dominance. So one allele is not completely dominant over the other one. So the heterozygote phenotype is actually a mixture, or is a blending, or is an intermediate of the two homozygote phenotype. So here, for example, I have a red flower and I have white flower. So if I mix them, so I will have actually a mixture between the two, which is pink. So this pink or this phenotype, you cannot see it in the parents, but we see it in F1. So this is the parents. So if you mix F1 with F1, then you will see what we will get later. Okay. So here, for example, 4 o'clock flowers. These flowers are called 4 o'clock. Okay. So these flowers, they are red. So let's say the red one is RR, the white is WW. So the heterozygote, when you mix these two, it will give you what? It will give you pink. It's a mixture of the two alleles. So the pink one, so this one is homozygote for white, homozygote for red, and this one is heterozygote. Okay, so if you want to see the Punnett square, etc., so you can actually represent it like this, or simply you can put RR. So in this case, because since both of them they are dominant, this one we will be called R capital, and this one will be called W capital, because both of them are dominant. So if you have a flower which is sorry, I put in the center, R, R, you cross it with the flower W, W, what are you going to have? So you have this one, the gametes are R, R, so R, R. Of course, you are going to draw the Punnett square. The other one will be W, W. So what are you going to have? All your offspring, F1, will be R, W. R, W, both capital, huh? So, what will be the genotype? Is actually RW, 100%. And phenotype, what is it going to be? It will be pink, 100%. Okay, so this is the case of this flower. Remember, for pea plant is different. Pea plant, you have purple and white, and purple is dominant over the white. So you don't have that mixture, but this one, in this type of flower, we have this mixture. So now if you mix F1 with F1, what are you going to have in F2? So now look at F2. So what are you going to have? F1 with F1 means will be RW crossed with RW. So what are you going to have? You have RW here, and you have here RW. So what are you going to have? So R, R, RW, RW. Okay, why I start with the R here? We have to follow the alphabetical order. Okay, you cannot put WR. And here we will have WW. Okay, now what is genotype? Genotype, you have RR, you have RW, and you have WW. What is the ratio? This one 1, this one 2, and this one 1. So is 1, 2, 1. This is the ratio. What about phenotype? Phenotype, it will be, this one will be red, this two will be pink, and this one will be white. Oh, sorry, no space here. Then, what is the ratio? The red is 1, the pink is 2, and the white is 1. So, here also the ratio will be 1 to 1. So, this is what is represented here. So, this is what is written here. Okay, so this is, you have in F2, so F1, all of them pink. So this is actually the RW here, you have the gametes. When you cross F1 with F1, you have here RW, RW. So here you will have RR, red flower, RW, pink flower, RW, pink flower, and WW, white flower. So what you can see here in F2, what can you see? You can see that the phenotype of the parents reappear, and you have also the phenotype of the mixture. Okay, so this is an example of codominance or incomplete dominance. So here, incomplete dominance, we will see the example in human of sickle cell anemia. So sickle cell anemia is actually, this one we have actually, if you do remember, we have seen last year in chapter 17. So, if a person inherits both recessive alleles, so HBS, so recessive alleles for sickle cell hemoglobin, then he will have the disease. So if a heterozygote HBS, HBA will have the sickle cell trait, so mild syndrome or mild symptoms, of anemia. Okay, I will start first. What is sickle cell anemia? So sickle cell anemia is a condition where the shape of the RBCs, the red blood cells in the blood, this is the normal shape of the RBCs, and this is what we call sickle cell. It's like a crescent. It will be like a crescent. What caused the sickle? What caused the cells to have this shape? Okay, so the story starts in chromosome 11. So this is the list of chromosomes we have. So you can see here chromosome 1, chromosome 2, etc., chromosome 22, and XY. So from 1 to 22, how do we call these chromosomes? Do you remember? We call them autosomes. Autosomes. And the last pair of chromosomes, we call them sex chromosomes. Okay? Like this here, this set of chromosomes is a is a male because it has XY. If it is a female, she will have 2X. Of course, here you have 2 copies of one, 2 copies of two, 2 copies of each. Okay. So now what happened in chromosome 11? So the sickle cell disease is caused by a mutation in the hemoglobin beta gene found on the chromosome 11. So in the chromosome 11, we have a gene here which code for hemoglobin beta, because hemoglobin you have 2 types, alpha and beta. So there is a gene located here, code for hemoglobin. Okay, this one you don't have to memorize, don't worry. This one as well, you don't have to memorize. I just want to show you where the mutation happened. So you can see this is the chromosome 11. It was fully sequenced, and we know exactly where the gene is located. It's somewhere here where I show the arrow. That is the position of the gene. If you look at the sequence of that gene, by the way, you can see here the sequence of the chromosome 11 is 134 million base pair. Okay, and we know them all. We know all the sequence. So here we look at the sickle cell anemia. If you look at the sequence of the gene, what are you going to find? You are going to find, okay, so this is will be your chromosome. And here, let's say the gene starts from here, and there is a certain position. Normally here you have G, A, G, and here you have C, of course T, C. So, and of course you have the sequences here, but we just want to mention this one because there where the mutation will take place. Okay, so when you have your mRNA, what are you going to have? In your mRNA, this is actually the template strand, and this is the coding strand. So when you have your mRNA, the RNA polymerase will bind to this strand and will copy it. So we will have G, A, G. Okay, of course here you have the other sequences. Okay, this is your mRNA. Now your mRNA will go to the cytoplasm and the ribosome will start reading, isn't it? When the ribosome read and produce the proteins, so you will have the different amino acid, the first, second, different amino acid. When the ribosome reach here, the amino acid will be actually glutamine, glue. Okay, then it will continue. This is the hemoglobin, or actually the globin, to be more precise, globin. So now this globin protein, later on it will combine with the heme. You remember the heme? So the globin will combine with the heme and it will make what we call hemoglobin, or we can simplify it by a short letter like this, hemoglobin. I can show you the picture here. I have it. Okay. So so it's actually here. You can see the red one. So this is the hemoglobin. So actually the hemoglobin in red blood cells is made of 4 proteins. 4. You can see 1, 2, 3, 4. So these 2 are beta globin, these 2 are alpha globin, beta and alpha globin. Okay, so this one is the heme, the red one. So each protein will have a heme. Okay, so this is when you have a normal hemoglobin, it will be as shown in the photo here. Okay, now when you have this mutation, so what is the mutation? I will show here. So the mutation here, that's this A become T. If the A become T here, what will happen? So here it will become A. And if it become A here, so the mRNA here will change. So you have G and A. What do you have for A? You have a U. And here you have, of course, so you can see a single mutation. A single mutation in this coding strand will cause this to happen. So now, okay, the ribosome. When the ribosome read G, U, G, actually the amino acid will not be glutamine. You can check actually your codon chart. It will be actually valine. So here the amino acid will change to valine. So when the amino acid change to valine here, okay, here you have to talk about the physical structure of the amino acids. So when the amino acid change to valine here, what will happen? Actually, the protein will change. The proteins will change. So what will happen? The protein, the shape of the protein will change, and the proteins, the hemoglobin, will stick to each other. When this sticks to each other, they will make like a rope. A rope. So now imagine the cell, and all these proteins inside the cell, the RBC, will make like this rope. So this is what we give. This is what give the. So here you have hemoglobin will clamp, and when this happen, so when this happen, what the cells become sickle. We call them sickle. So means like they have this crescent, and you can see. I don't know whether you can see the rope shape here of the hemoglobin inside the RBC. So when this happen, these cells cannot pass smoothly in the blood vessels, especially the small capillaries, and they will clamp. When they clamp here, what will happen? Means this part of the body, of the tissue, will not receive any blood. Imagine this happen in your brain, or happen in the heart, or happen in any organ. What will happen? In the brain, for example, it will cause stroke. If it happens in the heart, it will cause heart attack. It can happen anywhere. Okay? So, you can see this animation. I put it in the PowerPoint, so to see how exactly it will work. So, now, if you receive normal hemoglobin, you will be called HbA, HbA. So, you are normal. Your hemoglobin is normal. If you receive one copy with mutation, one copy is normal, you will call HbA, HbS. So Hb is here for hemoglobin, S is for sickle, A for normal. If you receive both copies with sickle cell anemia, means all your cells will be sickle like this. Okay, so I just want to mention here, or maybe later. Okay, how do we inherit that? Okay, so how is sickle cell anemia inherited? It appears like a recessive one, but actually is not fully recessive. Okay, so if your father is carrier, your mother is carrier, means your father is, I put here, if the father is HbA, HbS, and the mother is HbA, HbS. So, what are you going to have here? So you have HbA, HbA. Here you have HbA, HbS. Here you have HbA, HbS. And here you have HbS, HbS. So this one will suffer the more. Okay? So this one will be normal. HbA, HbA normal, but this one will suffer the most because all his cells are sickle. All his cells are sickle. But this one, these two here, so this one is heterozygote. Heterozygote means half of their cells will be normal and half of their cells will be sickle. Okay, so this will have the sickle cell anemia, they will have the disease, but these two, they don't have really the disease, but they have the sickle cell trait. We call it sickle cell trait. They will have sickle cell trait. So I hope this one is clear here. Okay, so this is what we can have. Okay, I remove this. Okay, so you have normal, HbA, HbA. So what is the phenotype? Is A. This one, sickle cell anemia. This one will have definitely the disease, and they don't survive because they will be dead by, the disease. So HbS, HbS. So the phenotype is the type of hemoglobin present is S. But this one, they have sickle cell traits, HbS, HbA, S and A. They have both hemoglobin. So the normal hemoglobin allele HbA is not completely dominant over the recessive one. So we are going to talk mostly about these people. Why the S is not fully recessive, or the A is not fully dominant over the S? Because these people here will suffer in certain condition. What are those conditions? When the oxygen level in the blood is low, then they will start suffering. Because normally the cells are the sickle cell or the cells with the mutated gene, they are also normal. But when you have this low condition, the cells will start to sickle. So people with sickle cell traits, what does it mean? Sickle cell trait, not with sickle cell anemia, means those with, okay, I write it here so you will not be confused. Sickle cell traits means those which are HbA, HbS, means they are heterozygote. So will develop symptoms of sickle cell disease under extreme condition. What are those conditions? Those conditions include an increased pressure in the atmosphere, such as scuba diving. When they start diving, they will start suffering. Means their cells will start to sickle and they will have less oxygen, because when the cells sickle, means they will not bind to oxygen, so the person will have low oxygen in the blood. So when you have low oxygen levels, which can occur while you are performing a strenuous physical activity, when you exercise, you need more oxygen in your blood, isn't it? So these people, when they exercise, they will start feeling the symptoms of the sickle cell disease. When they are at high altitude also, they will feel these symptoms, or they start to develop the symptoms. And when they have dehydration, like athletes with sickle cell traits, they need to take precaution when performing. So staying hydrated is extremely important for them. Okay, so here you have some, but this one is not included. I just want to show you what is sickle cell anemia. What does it mean? When you have a sickle cells, what are the symptoms? So here, first, these sickle cells, when the cells sickle, they actually break easily. They start breaking. So they will break down, and they will clump and clog the blood vessels, and they will accumulate in the spleen. So how this will affect the organs? So here it will cause a brain damage. It will cause also, it may cause heart failure. It causes anemia when they start breaking down. Physical weakness. It can cause also damage to other organs like kidney, like other organs. So it can cause kidney failure, can cause rheumatism, can cause pneumonia in the infection in the lungs, can cause even paralysis if it causes brain damage, and can cause also impaired mental function. So this is actually why is it dangerous for sickle cell anemia. And I don't know if you do remember from last year, we have seen about malaria. Do you remember malaria? So we have seen that. Well, let me see. So let's say you have a population with HbA. You have a population, I mean in the same population, you take any country, for example, let's say Africa, there is some places where you have this HbS, and HbA, HbS. So in this a certain region of Africa where malaria spread, so what do you have? These people will die because of the disease. These people will die because of the malaria. Why? Remember, the malaria parasite is actually infect the RBCs. So usually the malaria parasites doesn't actually prefer the cells which are sickle. So they infect mostly the people which have normal hemoglobin. So who will survive? Only those people which are heterozygote for sickle cell, because there will be, so this is a kind of natural selection. Okay, so this one we have discussed last year, and that's all. So this is the end of my presentation. Hopefully it will be helpful. Wassalamu alaikum wa rahmatullahi ta'ala wa barakatuh. If you have any comments, please don't hesitate to ask or to comment in the group, WhatsApp group. Yalla, assalamu alaikum.